Hello. Today I'll be discussing a recent paper by Kagiata and colleagues titled Predictive Absolute Protein Folding Stability Using Generative Models from Protein Science. Here, the authors are interested in leveraging the evolutionary scale model, inverse folding, ESMIF, model likelihoods to assess delta delta G between wild type and variant sequences, using wild type sequence as input. First, they establish an approach assessing equilibrium to state folding proteins with ESMIF information to predict absolute stability. Then, they explored how different configurations of a protein affect model prediction. Finally, the authors show important limitations for stability prediction with their model, especially when larger and more complex protein stability is dependent uh, upon cooperativity between structural elements of specific folds. Their approach was benchmarked against 265 experimentally determined protein stabilities, showing performance with a mean error of 1.5 kcal per mole and correlation coefficient of 0.7. However, their success was limited within groups of small to mid-size two-state and single-domain proteins. Additionally, assessment of specific residues correlating with stability showed that valine and isoleucine highly correlated to global stability. Hydrophobic residues consistently correlated with global stability at the same extent as the total sum of likelihoods, which aligns with expectations for the impact of hydrophobic effect on protein stability. When evaluating whether the model could predict different stability between protein conformations, they found that T4 lysozyme E state was less stable than G state, in agreement with experimental results. However, a limitation in their model in the cases of both T4 lysozyme and cyclophilin A states was that they predicted a difference in stability between states that was a lower magnitude than what was described experimentally. In comparison to alternative machine learning models, here using ESM1B and FoldX's empirical force field, ESMIF consistently achieved the highest correlation with experimental stability compared to the other approaches. Overall, the authors showed that their model performed consistently better than alternative sequence-only machine learning models and standard empirical force fields. They showed that their model can achieve likelihoods for amino acid predictions that match closely to experimental results for small to mid-sized proteins up to about 150 amino acids with one or two states. They're limited by the multi-state proteins with folding complexities brought upon by increasing protein size. Nevertheless, future work involves finding ways to implement decomposition of large proteins for analysis, improving training data size and including more complex proteins, implementation of side chain information, and other protein biophysical data. Thank you for listening.